Okay, so hello everybody. Welcome and thank you for joining us for another round of the online Cell Size and Growth seminar series. Today it's a real pleasure to have two talks from people who are going to talk about the kind of extreme end of the size spectrum and how that relates to different aspects of cell biology and cell function. We're going to start with Ben, who's going to talk to us about cannibalistic supergiant cells. So Ben, without any further ado, would you like to share your slides and get us started?
Oh, absolutely. It looks like I have to send a request to share now for some reason. All right. Well, as I'm getting this set up, just thanks for the opportunity to talk to this audience. I'm guessing I'm going to be a little bit outside of the norm for everyone, so I'm going to try to spend a good amount of time in intro and setup for all of this. But I'm really excited to hear your questions at the end of this talk. So without further ado, I'll introduce the star of our show. This is Euplodes. This is a single-celled organism, a ciliate that, as you can see, is walking across a surface. This is the bottom of a flask, and more or less how I encountered this cell in a field sample. And for me, I look out at all these fantastic microbial eukaryotes, these protists out in the world, and I really want to understand how it is that they control these kinds of complex behaviors. So the core goal of my lab is to study complex cellular behavior, and we usually start by putting cells under the microscope and quantitatively characterizing movement patterns as a behavioral readout. We use then perturbative experiments to get at molecular and physical mechanisms underpinning behavioral control. But we want to understand, not only what cells do in the lab, but what they do out in the world. So we have an appreciation for naturalistic behaviors, the environmental context of behaviors. And finally, we want to put all of that understanding together and with comparative approaches to understand something about how cellular behavior evolves. And I'm going to talk a bit more about this sort of comparative side of things when we get into the heart of this talk. Okay, so if like me, you're interested in sort of general principles of cellular behavior, it can be a bit overwhelming to look at the incredible diversity of form and function among eukaryotic cells if you look out in the world. So this is a tree of eukaryotes. Just to orient you, if you can see my cursor, animals are down here in this little corner. Okay, so there's this challenge in finding common principles among all this complex diversity, but there's opportunity too. So some opportunity is that despite all of this diversity, there are actually shared physical constraints that many of these cells operate under. They're relatively close in size, although there is quite a wide size spectrum. And they also have this shared common ancestry, which you can see in the convergence of all these branches, reflected in the fact that we have shared cellular components, molecular components of the cell, despite all this diversity. Okay, we also have this opportunity to find perfect or great examples among this diversity to highlight our particular biological questions of interest. And so for me, with this question of how cells control complex behaviors, I want, a few boxes checked. I want a cell that has this complex behavioral repertoire that I can study in the lab. And also importantly for these, comparative approaches, I want to have behavioral and morphological diversity across species. So Euplodes, I think, is a really fantastic system where we can use its unique biological features to shed light on biological questions, which would be hard to address and study in a lot of traditional systems. So here's this Euplodes cell. You can find them in aquatic environments all over the place. The cell's about 150 microns long and has this highly polarized structure. They really have this animal-like body plan, and this is very useful for the work that I do or the work that my lab does. So there's a front and a back and a top and the bottom of the cell. Off the front of the cell, this fringy little structure here, this is part of a structure called the membranellar band or the adoral zone of membranelles, and it's a ciliary-based structure. And it beats those membranelles in a coordinated fashion to generate feeding currents that it pulls in under normal conditions, pulls in small bacteria and small other protists that they feed on. So part of the feeding apparatus of the cell. And off the bottom end of the cell, there are these leg-like appendages. So, those membranelles are ciliary structures. These legs also are ciliary structures. So if I were to take a cross-section just below the cell membrane of one of these little legs, you'd see that each leg is a bundle of this highly ordered array of cilia. Okay, so it's a ciliate. It's not covered in cilia, but it has these bundles of cilia. And so, early protostologists found these cells. They're large. You can find them in a lot of different environments. And they saw this kind of complex animal-like cell structure. And so here is a diagram from a Berkeley thesis from the early 1900s where we've drawn this cell. This is that adoral zone of membranelles, that membranellar band. And they could see all this internal structure in the cell just by light microscopy. And so they looked at the legs of this cell. These are the bases of all those little legs called cirri. And they saw these fibers interconnecting among the legs of the cell. And they thought this must be something like a rudimentary sort of nervous system. Of course, we know that's not the case, but they weren't imagining things. So here is a visualization of the microtubule cytoskeleton of the cell that's visualized by confocal microscopy. And so you can see these bundles. They're not nerve fibers. They're bundles of microtubules, in fact, that are interconnecting among the cells. So the cell has this really complex yet highly stereotyped architecture. It's really animal-like. Another thing I want to point out in this diagram that will be important coming up pretty soon here is this structure right here is actually part of the mouth of the cell. So under normal conditions, they're filter feeding, they pull in bacterial prey, they catch it here and engulf it in that mouth structure. Okay. So in addition to this morphological complexity, they have behavioral complexity. So these cells, live out in the world, they hunt. And so here are some experiments where we're looking at different cell movement patterns in the presence of different prey conditions. So this is Euplodes eurystomus. In this case, that's the species of Euplodes, and these clammy are the prey. And if we change the concentration and distribution of prey, we can evoke these different behavioral responses of cells. So these different colors are just trajectories of cells running around in their enclosures. So with dense prey, the cells aren't moving much. They're mostly just sort of standing still, filter feeding. With patchy prey, maybe most vividly illustrated by this yellow trajectory here, they're executing these local search patterns. So they have these evocable different behaviors in the presence of different prey. And all of that, different movement, different behavior is driven by the movement of these leg-like appendages of the cell. So a lot of my lab is trying to figure out how these cells actually coordinate these leg-like appendages. And so this is a cell that's imaged under high-speed microscopy. You can see the cell sort of reorienting, running off in a new direction here. It's doing all this while maintaining contact with the cover slip. And one of the things that we found, just to skip ahead, is that this internal system of bundled microtubules is actually important for mediating coordination among the appendages. And this is a bit reminiscent of what we see in a bunch of other ciliated organisms, other protists, and then also in multiciliated cells in the human body. So there may be these sort of shared principles. But what I really want to emphasize for this talk is that there's this integral relationship between cell architecture and cell behavior in this cell. And I think this is really a strength of the system. And so as we, try to look a bit comparatively to understand something about the evolution of cellular behavior, we're assembling a bit of a zoo in the lab. And so here we have for example, three different species of Euplodes. They have different cell movement patterns, and they also have these different architectures of these bundled microtubules. And so we're working on understanding how it is that modifications in the cell architecture, the cytoskeletal architecture in particular, might give rise to these different kinds of behavioral capacities or different movement patterns of the cells. So this kind of comparative work takes us out into the field to look at different species of Euplodes, the different environments they live in, and how they navigate those different environments. And so today, the real meat of this talk is going to focus on an adventure that stemmed from this work of going out into the field and finding new species of Euplodes. So I was lucky enough early in my career to establish a field site on the island of Curacao, which is just north of Venezuela in the Caribbean here, this tropical island. There's a field station where you can go do research, and I've sampled all around the island, found a bunch of interesting protists. But one place that I hadn't yet sampled in all of these sites that are available to us was the lab station itself. So right over here in this corner of the field station, there are these water tables that take in water from the ocean and then supply different tanks that house different critters that people are working on. So I decided, where am I going to find an interesting heterotrophic protist? Perhaps on the filter of this water table system. And so I scraped this water table filter and put that material into a flask and throw it in the back of an incubator and kind of forgot about it actually for a couple of weeks. And I was excited to find, after a while, I looked in and there were these Euplodes around. And after getting them into culture, so from a clonal isolate, I grew up these cultures of cells, and I found that every once in a while I would see these really huge cells intermixed with these smaller cells. And I became very fascinated with these organisms. And so here we have a video of what's from a clonal population of cells. All of these blobs you see here are Euplodes, and you're going to see these smaller cells are mostly going to be hanging out, filter feeding, doing what Euplodes tends to do. But quite strikingly, in addition to this morphological change, I think you'll appreciate this behavioral change in this cell as well. So here we have this cannibalistic predation event. And so here I got really excited seeing this because we have this morphological transition that's associated with this dramatic behavioral difference where you have a cell that transitions from this filter feeding lifestyle to this predatory, cannibalistic sort of mode. And I'm hoping that we can build this system as a unique window onto ecological evolutionary developmental cell biology. And I'm going to talk more about what that means coming up here. Okay, so we had these cells that would ephemerally appear in the culture. And just to give us a bit of context for this cannibalistic type of behavior, this is not unprecedented among eukaryotic cells. So there are other ciliate species, Blepharisma and Tetrahymenovorax, probably most famous, that have these predatory transitions where cell size increases and you have this change from more of a filter feeding sort of lifestyle to this predatory lifestyle, carnivorous lifestyle. It's described in the literature often. But we also see cannibalistic cells broadly across the tree of eukaryotes. These, of course, are all independently evolved, but in Dictyostelium, you may be familiar with this social slime mode. There is a cannibalistic lifestyle that some species exhibit, and also in animal cells, right? In cancer, cell cannibalism can happen as well. So, okay, so it's this sort of more general phenomenon that we see in these different organisms. Okay, so back to our Euplotes species. Okay, so um, I worked with collaborators in Patrick Keeling's lab and identified, okay, so this is in fact a previously undescribed species of Euplotes. We named it Gigatrox in reference to the giant fierce cells that we have. Um, and it's a new species of Euplotes, but it looks more or less like most Euplotes species, where we have these cirri on the bottom of the cell, this adoral zone of membranelles, and so cell architecture is like other species of Euplotes. But a key difference here is this dramatic phenotypic polymorphism. So we have these supergiant cells that we saw. These are just electron microscopy images of the different morphs. Supergiants here, these normal morphs. And then we also noticed the appearance of these winged sort of defensive morphs. So in other species of Euplotes, there is this compensatory phenotypic plasticity that's exhibited by some species where in the presence of predation, cell bodies get too big to fit into the mouths of predators. And we see that sometimes going on here. But we really wanted to study this supergiant transition in a bit more detail. Okay, so there is this morphological change that these cells exhibit. Normal morph compared to a supergiant. These are sort of quantifications of different morphological aspects of the cell. Obviously, the cells are much longer. This is aspect ratio, so the supergiants are boxier. They also have this extended adoral zone of membranelle. So they actually are extending a portion of the cell to accommodate these larger prey items that they feed on predatorily. Okay, so in addition to this morphological difference, this change in cell size and shape, we have behavioral differences that are quite striking. So of course, the supergiants exhibit this cannibalistic feeding, but the movement patterns of the cells are actually quite different when you compare them to normal morphs. So here in these different colors are just trajectories of cells freely behaving in a flask. And with the normal morphs, you can see this diversity of different walking movements. So these straighter lines are walking movements, and these little curly cues actually correspond to helical swimming of the cells. So Euplotes, I study mostly the walking behavior, but they're quite good at swimming as well. With these supergiants, quite noticeably, they tend to exhibit these circulatory walking movement patterns. So these are just some quantifications over here of different aspects of cell movement. The supergiants in red here, they're moving faster. They have this characteristic angle that they're circulating when they're walking. But one thing that we noticed importantly is that I almost never saw them swimming spontaneously. And I wondered perhaps if there was some sort of phenotypic trade-off where they have this rapid movement on surfaces, predatorily feeding, but perhaps they're limited in their ability to swim. And indeed, when I put them in little enclosures and gently disturb them from the surface that they're walking on, this is a mixed population of normal cells and supergiants. When the supergiants are knocked from a surface, they sort of tumble a bit helplessly, and the normal morphs you'll see will swim away quite well. So if you look at the swimming patterns of these cells, these are just cell trajectories, and we've centered the mean position along all these trajectories, and we find indeed these supergiants seem to exhibit this sort of trade-off that perhaps limits their dispersal. So they really seem to be bound to this sort of surface-based lifestyle. They're feeding on surfaces. As far as we can tell, they can't really feed while swimming. Okay. So this morphological behavioral transition associated with perhaps behavioral trade-offs in terms of dispersal capability. So all of that work had really done while taking advantage of these ephemeral blooms of these supergiants that I could find in cultures, but we wanted to get a better handle on controlling cell growth and to try to dial in the conditions under which these supergiant cells actually form. And so we tested a bunch of different conditions, and the breakthrough really came from dialing in specific media formulations. So we feed these cells basically different teas from different seaweeds and different grains that grow up the prey items in the culture that the cells then graze down. And what I found as I started to play around with media is there was sort of a sweet spot. This is RA, this is this red algal media, where if I gave these, moderately low nutrient availability for the cells, these are just growth curves of the overall population of cells in cultures. I saw this bloom of supergiants that tended to correspond with when the population just sort of hits this stationary phase of growth. Although there was sort of a sweet spot in media where if I gave the cells enriched media, high nutrient concentration media, I'd see still this, logistic like growth of the population, but no appearance of supergiants. And maybe, unsurprisingly, if I give them just artificial seawater, dilute media, I don't see the emergence of these supergiants. And so another thing we noticed about these supergiant blooms is that the cells supergiants appear, they tend to not exceed more than a few percent of the population. So about 5% of the population is the highest abundance in terms of fraction of cells that are supergiants. And they relatively quickly sort of disappear. And so the supergiants can actually grow and persist as supergiants in the culture, but can also undergo, so they can divide and persist as supergiants, or they can undergo these asymmetric cell divisions that eventually bring them back to this normal morph phase. And it turns out that supergiant persistence does depend on prey conditions as well. So if they have their conspecifics as prey items, these large prey items that they're feeding on cannibalistically, in the case of all our cultures, they can persist for up to a little over a week. But if prey items, small prey items, so bacteria that the normal morphs are feeding on, or we can also feed them small algae, if those small prey items become abundant, or there's just no prey around, we just put them into seawater, these supergiants will very quickly, within a day, revert back to these normal morphs. So supergiant formation and persistence is sort of tuned to the environmental conditions. Okay, so we wondered a bit about, there was this interesting characteristic of the cells sometimes forming these supergiants when the cells hit this sort of stationary phase, but in other times not. We wondered if there was some sort of, in addition to the external cues, some internal state dependence on supergiant formation. And so when we picked individual cells, so we started them from a supergiant morph or we started them from a normal morph, we found that populations started from the supergiant morph actually tended to not as rapidly and not as frequently form supergiants later. And so we thought, okay, maybe there's some sort of internal control in addition to these external controls. So to start to get a bit of insight into perhaps how this morphogenetic transition is actually regulated, we actually collaborator Daniela Giannotti and Patrick Keeling's lab picked individual cells and performed transcriptomic analysis on these different morphs. So we have here PCA plot of these transcriptomic results of gene expression for these different morphs. So we have supergiants here, normal morphs here, reverted cells, so that were previously supergiants and had become this by eye normal morph morphology. We also added one more type of cell that we could find to the mix. So there are these intermediate cells. So these cells look a bit like this, and they're just morphologically defined. They are not as big as supergiants, but they are bigger than normal morphs, and they don't have this sort of standard normal morphology. So, one thing that's tricky about just this morphological criterion is we don't know if they're maybe becoming supergiants or reverting from having been supergiants. But anyway, what we find is that broadly, in terms of patterns of gene expression, we find these distinct gene expression profiles corresponding to these different cell types that we identify, normal, supergiant, and reverted. And so we think this is basically evidence of this morphogenetic cycle through patterns of gene expression. And so there's a lot that we'd like to dig into in terms of molecular control. We're really at the early days of this organism. It's a wild organism that we took in from the environment. And if any of you have worked with protist transcriptomes, unless you're working on a very well-established, more traditional model system, a lot of the genes will come back without sort of any annotation. So it becomes a bit tricky to work on this. But one thing that we did notice when we looked at, in particular, supergiants versus normal morphs, is one of the most differentially expressed genes was a CDK in normal morphs. Looks most like CDK2, perhaps CDK2 ortholog. And so I just recently, this is very late breaking data, but I'm excited to at least show this preliminary result. So I just had a rotation student, in fact, who tested a bunch of CDK inhibitor cells and saw, I think, what's quite an interesting phenotype. So we have our normal morph here, supergiant morph here, and with the CDK inhibited cell, we see the appearance of these weird morphologies where the cells are big, they're not quite supergiant size, they have this tend to have this weird extended portion of the front of the cell. It's a bit hard to tell, so these cells are viewed sort of top down. This is one of these strange morphologies viewed in profile here. And so we think, at least very preliminarily, that this is, evidence that there is some sort of interesting non-trivial cell cycle regulation that we are hoping to follow up on, and perhaps can find some link to this supergiant cell morphogenetic transition. And so one thing that this sort of reminded me of, again, I'll just say this is a very preliminary result, but it got me thinking about developmental mechanisms and evolutionary developmental biology in animals, whereby one sort of general class of mechanisms for morphological changes is called heterochrony. This is basically the changing in the timing of a particular developmental process that can give rise to different kinds of morphologies over evolutionary timescales. And so one somewhat famous example is the rescaling of dog skulls through the differential timing of different developmental processes that is giving rise to different dog morphologies over the course of domestication. And I got to thinking a little bit about, drawing, more substantive comparisons to what might be happening in cells. And I was very much inspired by a recent paper from the Writer Lab led by Asemil Choksi, where they looked at multiciliated cell developments in animal cells. And what they found is really there's this rewiring of the standard cell cycle that gives rise to this different cell morphology in this differentiation process. And so I'm wondering if maybe we see something a bit reminiscent, of course this is very speculative, in our supergiant transition, whereby we have alteration of the cell cycle, repression perhaps of the CDK2 that extends perhaps this G1 phase of growth, that can give rise to these dramatically different cell morphologies and associated behaviors in our euploides system. Okay, so that's very speculative, very, very preliminary data for now. But I think that this system could really be an exciting opportunity to take this sort of evolutionary developmental biology approach to studying a developmental process in a cell that might help us understand the elaboration of different cell morphologies and behaviors across evolutionary timescales as well. So just to summarize, what we found with these supergiants is we think that everything that we're seeing is sort of consistent with a bet hedging strategy, where these normal morphs are undergoing this stochastic transition to this supergiant morph in the presence of dwindling small prey items. And so with that, I'd like to just wrap up by saying I think euploides could be a powerful system for understanding the evolution of cell behavior and morphology. And more broadly, across ciliates, we have this integral relationship between cell structure and behavior, whereby modification of cortical architecture, cytoskeletal architecture, can give rise to this incredible diversity of form and function among these different types of ciliates. So with that, I'll thank people who helped with this work in many ways, and then also thank you for your time and attention.
Amazing. Thank you, Ben, for what was really a fantastic and really enjoyable talk. I'm going to remind everybody, if you have questions, please post them in the chat and we can spend the next 10 minutes doing Q&A. I'm going to start, and I wanted to ask you whether, across both developmental programs and evolutionary time, there's this quite strong correlation between size and ploidy.
Yep. Yep.
As individual organisms change their ploidy, they
Yep.
typically increase size, and endoreplication is a way that some cells get massive. So
Absolutely.
do you see that? Have you been able to look at the ploidy of these cells?
So would love to. Okay, so with ciliates, it's a little bit complicated. I don't know if you know how much you know about their nuclear biology, but they have a macronucleus and a micronucleus. This is all ciliates have this, differentiation where the micronucleus is basically acting as sort of a germline, transcriptionally silent, and the macronucleus is sort of the somatic genome. And so within the macronucleus, you have differential ploidy of different genes. So you can up to have, up to a thousandfold copy of specific different genes. And, so ploidy is a bit complicated to answer straightforwardly. For what it's worth, just in terms of very, gross evaluation of this kind of question, under normal supergiant formation, we do have what looks to be scaling of the nuclear size with cell morphology that you would expect if you would have the same scaling relationship as for the normal morphs. But I do love the idea that you have, a different genome within these supergiant cells, working on pursuing that. But I don't have anything more to tell you beyond that. The size of the nucleus itself scales proportionally with the size of the cell.
Cool. That's
Yeah.
super interesting. So next up, we have a question from Kiki.
Hello. Should I read the question aloud?
Yes, please.
Sure.
You mentioned that supergiants can eat prey other than members of their own species.
Uh-huh.
Do you believe that cannibalism is actually an important part of the life of this species in the wild?
Yep.
Or is its preference in experimental conditions a consequence of the unrepresentative rarity of large prey of other species? I
Yep.
suspect that cannibalism is less of a specific adaptation and more of a consequence of a lack of distinction between own species and other species prey, which might occur much less in the wild simply because the latter is much more abundant than in the lab.
Yep. That's a great question, and I think sort of spot on. So I don't believe that this phenotypic transition is, tied very specifically to cannibalism, but I don't know anything about the natural context under which these transitions take place. The one sort of complicating aspect of this, so I do also think of it as, okay, these cells are starting to get hungry. There's no small prey around. Let's find some other prey source. Let's eat big things. One complicating aspect of the transition is that it does seem to occur after these phases of rapid growth. And because it has that sort of cell growth dependence on the transition, recent cell growth, you could imagine that this could, in natural context, actually take place in the context of blooms, of ephemeral blooms, and in which case they could plausibly be even primarily cannibalistic. I just don't know enough and I would love to go back and keep sampling. I would love to find supergiants in the wild. I think it would be very hard, but I really don't know anything about the natural environment under which this
That
is.
is a reasonable argument
Yep.
in that in a bloom situation where you get a burst of exponential growth
Yep.
before carrying capacity is approached.
Yep.
You could there there could be some kind of incentive to switch to basically stealing energy from other members of your own species.
Right, right, right. And in that case, I mean, maybe from clones. So they are able to prey or predate. They eat about, one prey every 10 minutes. And after they revert to the normal morph phase, they can form, I think the most we had is up to 18 cells, 18 normal morphs from a single supergiant cell. So it could be, rapidly accumulate your resources and then go disperse elsewhere after that. But hard to know. Yeah.
But I also wonder whether that whole growth curve dependence
Mhm.
is actually analogous to a dynamic that would occur in the wild, or whether
Mhm.
it's simply an experimental situation that activates
Yep.
an adaptation that primarily exists for more conventional
Yep.
predation on other species.
Yep. I think that could absolutely be the case. That's a nice idea.
Thank you for your patience.
No problem.
Next up, we have a question from Jan.
Sure.
Hey Ben,
Hey.
nice to see you again, and nice to
Sure.
see that you're continuing to do such interesting
Yeah.
work. And thanks for the talk and joining our series.
Yeah.
So I was wondering how they divide.
Yeah.
Like, do they
Great.
just do binary fission?
So it's a bit more complicated. It's so fascinating. Okay, so what happens is the first visible sign of cell division is that they'll actually start growing more legs. So these cirri, they're a subset of the cirri that they're templating the growth of new cirri from. So they grow more legs, those migrate to where they need to go. And the whole division process is one sort of elongation. So, along the front to back axis of the cell, they're elongating and they maintain motility during almost this entire process as well. So the cell maintains function as it's dividing. What's happening internally, would love to know. Don't know much about that. But it is binary fission, but it's this cell elongation process. So they maintain polarity the whole time, and they just inactivate cell movement just before the two pieces run apart. So
So
fascinating.
is a way of like, I don't know, maybe becoming a supergiant somehow like inhibiting the division
Yeah.
process and then like doing kind of like an endocycle, like along the lines of Matthew's question. Uh,
I think that might be, exactly what's going on here. But there's a ton more that we have to do to really prove that out, and there could be a lot more complexity in terms of
Yeah. I
which
mean, it's just
branches of the
hard
cell
to
cycle.
like, I guess, monitor them for that long, right?
Yeah,
Like how long does it take them to double in mass
yeah. So it's a good
in the
question
lab condition?
for the super giants. Like I said, they're sort of
No, no,
feeding.
for the normal one, for the
Oh, for
normal
the
one.
normal ones, it's like 8 hour division time.
Oh, really?
Yeah,
Oh, that might be doable then.
yeah.
Like as you change the media to this like poor media, you might be able to film it long enough and just see what it does.
Oh, yeah, absolutely.
Yeah.
Yeah, yeah.
Um, yeah, so that's the other question I had was like that when you switch to what you call poor media, does it just grow slower? So it's like a slower growth rate. And it's kind of interesting that like it would become bigger when it's growing slower. That's
Right.
a little bit counterintuitive.
It is, but what seems to happen is that I don't know. I have no idea about how this is controlled, but the sort of supergiant transition, it seems to be activated and or stabilized in a very small subset of the population. So if you just look at sort of normal population growth, yeah, they grow slower with lower media richness. Um,
Yeah.
but you're still getting this, growth and division of cells, and then stochastically you'll get this small subset of the population that will undergo this supergiant transformation. And what seems to be the case, this is again from somewhat anecdotal evidence, there are more cells than become actual supergiants that have this intermediate phase where they extend that adoral zone of membranelles, and then those cells can feed cannibalistically. And so I think what is required is this rare successful predation event to stabilize that transition. And then the supergiants themselves, they grow quite rapidly. So,
Yeah.
within a few hours, they can actually reach this really huge size once they start feeding effectively. Yeah.
Okay, cool. That gives me a bit better sense of the dynamics of this thing.
Sure.
Awesome.
Sure.
Thank you.
Yeah.
All right, I think we've got time for one more question, so we can do Teemu's question.
Hey, beautiful talk. My question isn't super well formulated in a sense, but can you
Sure.
just talk a little bit more about this supergiant formation and how that might enable them to eat different size prey? And
Yeah.
are there a lot of small prey in their environment normally that like
Yep.
maybe that's part of the reason for the
Yep.
yeah.
So standard euplotes, so across most species, they're bacterivorous, and they also eat small protists. So they're feeding on these small prey items, and they capture those small prey items by actually drawing them in with this feeding current. And so these supergiants have a very different mode of feeding where it seems that they need to actually run over the top of a prey item to encounter it. They're not trapping things in the feeding flow. I haven't done careful measurements of the feeding flows, but I suspect that the flow of the supergiants, in part just because the cell body is getting so bulky, they're not shaped appropriately to capture small prey effectively. And so part of what's happening is the cell it's the legs, the cirri get longer so they can accommodate a larger prey item. And then importantly, this adoral zone of membranelles, this AZM, this band, extends quite a bit, proportionally even to the size of the cell. It's extended even more so to accommodate for these large prey items. So it's really a physical change in cell morphology that just allows them to catch these bigger prey items for it to get into the cytostome, the mouth of the cell.
Got it. Thank you.
Yeah, yeah.
All right, there's one final question which we can do, if we can do it quickly, from Kiki, and then we'll move on to the next talk.
Less seriously, can normal-sized individuals escape cannibalism by supergiants simply by swimming away?
Yeah, so they pretty effectively get away. So they have these mechanosensitive cirri on the back of the cell, and that allows them to get out of the way. Although I'm surprised by how effective the supergiants are at actually capturing them. I would love to learn more about that behavioral interplay, because I don't know much about all the different mechanisms they can use to sort of escape. There's also this transition to these winged morphs that are too big to fit into the mouths of the cannibals. So.
If the normal ones can swim and the supergiants cannot, it seems that they have a rather obvious solution.
Indeed. But yeah, they don't all start swimming. So it's not something where, suddenly they realize they're under attack and they're all going to flee.
Okay, well, fantastic. Thank you.
In an evolutionary context.
Sorry. All right, well, thank you everyone for your questions. And thank you, Ben, again for a really wonderful talk. Next up, we have Teemu, who's going to talk to us about some other massive cells. So Teemu, if you'd like to share your slides, it's over to you.
Okay, you see my slides?
Looks good.
Wonderful. Great. First of all, thank you to the organizers for inviting me. I think I presented in this seminar series now. This is my third time, and unlike the last couple times where I've talked about details of mammalian cell growth regulation and fancy measurements for that, I'm going to completely switch gears and talk about phytoplankton. But hopefully this is still relevant for the cell size audience, because I'm going to be focusing on size and density regulation in these cells. So really quick primer. I know many of you work on budding yeast exclusively or mammalian cells. First of all, phytoplankton is basically the same thing as algae. I will be using those two terms kind of exchangeably. They are the primary producers of the ocean. So roughly speaking, half of all the global carbon fixation on our planet is done in the oceans, and that's done by phytoplankton species. They are very genetically diverse. We're not talking about one species or just one branch of the tree of life. And most of the species are actually very understudied. Like the previous presentation we had was not a phytoplankton, but a beautiful example of a marine single-celled organism that most of us probably didn't know about, because there's just so many of them and they haven't been studied. Now, there's a lot of interest in phytoplankton for a couple of different reasons. One is that they can be used in various kind of biomanufacturing, but more importantly for my talk, it's that the sinking of the biomass that has been formed by phytoplankton, especially the carbon that has been fixed into the phytoplankton, drives the ocean's biological pump. So the biological pump is essentially the process where carbon sinks from top or surface ocean towards deeper ocean, and some of that can eventually be sequestered for hundreds or thousands of years that way. Now, marine phytoplankton come in all kind of sizes. The size ranges are pretty crazy actually. So I have an approximate volume and dry mass scale here for you, where phytoplankton are classified into picoplankton, nanoplankton, microplankton, and macroplankton based on their size. And to give you some idea of what kind of species we're talking about, there's species like Prochlorococcus, which is technically a bacterial phytoplankton, but it's one of the smallest cells that is widely studied. It's way smaller than your typical E. coli, and it's super, super abundant. Probably the most abundant species on our planet. Then there are more better known organisms for a lot of you. Potentially E. huxley is an example, or Chlamydomonas is the most studied model system for phytoplankton. These would be nanoplankton in their size, so the size of a budding yeast cell or small mammalian cell. And then there's this micro and macroplankton, like this Noctiluca species studied by the lab of Manu Prakash, which is a beautiful example of a cell that can sink tens of meters in the ocean, then swell up by taking up tons of water into its essentially cytoplasm, or this kind of interestingly shaped vacuole that forms most of the cell. And that allows it to change its buoyancy, and the cell can start floating upwards for tens of meters. And its life cycle involves this kind of vertical long-distance migration based on buoyancy. And finally, you have this supersized phytoplankton, such as the Valonia ventricosa, which is a centimeter or more in diameter, a single cell. Now, the reason these size scales are interesting is that the gravitational sinking velocity of a cell is proportional to its radius squared. So the bigger the cell, the faster it's going to sink, or float if it's lower density than the water. And in my talk today, I'll be focusing on the sinking of single-celled phototrophic marine pico and nanoplankton. So I'll be only talking about the smaller size scale, because these are the species that are very abundant, and also these are the species where we have some measurements that work quite uniquely on them, but we can't do the measurements on all the really big species. Now, gravitational sinking is really the topic of today, and the question we're trying to understand is how do phytoplankton regulate their sinking in response to environmental changes? And the key thing to understand is that there are various kind of gradients in the ocean. So obviously there's more light towards the surface, so these photosynthesizing organisms essentially have more carbon available through photosynthesis when they're closer to the surface. Deeper down in the ocean, you have all kind of gradients appearing. There's salinity gradients, pH gradients, and so on. But most importantly for us, there are nutrient gradients for nitrogen, phosphorus, and so on. So there's essentially a eco-evolutionary pressure for phytoplankton to set their vertical depth in the water column. And plankton cannot, especially these small plankton, they cannot fully set where they are. They are kind of wandering partly based on ocean currents, but they have some control over their vertical position. And their vertical movement can take place by two different mechanisms. One is the gravitational sinking. So this is essentially a passive process that is set by the size, density, shape of the cell, and potential aggregation into larger aggregates. And then there's cell motility. In my today's talk, I'll just focus on gravitational sinking, and we'll only focus on species that don't aggregate significantly together. And the nice thing about this is that many of you might know the Stokes law, which essentially describes the sinking velocity of a, roughly speaking, spherical particle, and how that relates to its size and its density. So if you can just measure cell size, cell density, and have some qualification of its shape, you can actually derive the exact sinking velocity of a particle in a given fluid. And we can do that. So we can measure cell volumes using the Coulter counter, which is an electrical impedance-based volume measurement, and we can measure cell masses using the suspended microchannel resonator. Now, just very briefly, in the case you're not aware of, the Manalis lab here at MIT, where I work, has developed the suspended microchannel resonator, and this is essentially a vibrating diving board type system where the vibrational frequency of that cantilever is proportional to its total mass, like on any vibrating system. And we can flow cells through a microfluidic channel within this vibrating cantilever, and the change in that resonance frequency is just our measurement of point mass. It's very precise, and because we know the volume of cells from other measurements and we know the density of the fluid, we can then also calculate the density of the cells. And what we've done in a previous study with the Polt's lab, who used to be at MIT, is currently in Vienna, we've just scanned through a medium number of species from across the tree of life. These are all single-cell phytoplankton. We mapped out their typical volumes in high-growth environment, their densities, and their sinking velocities. And what you'll notice immediately is that you'll actually get a couple orders of magnitude of range in sinking velocity shown here on the right. And if you correlate sinking velocities with cell volumes, you'll get a very good correlation over these very large size scales. And the way to think about this is really that there's only so much that the density of a cell can vary. So over very large size scales, you can really understand the sinking of a particle just based on studying its size. And this is how really a lot of ecological models are considering sinking. You consider that there's a certain amount of cells or particles of given size, and the size gives it a certain sinking velocity. What is not normally considered in most ecological models is that sinking velocities may not be constant over time or when environmental conditions are changing. So if you look over a little bit narrower size ranges, you look at a specific species, it might still be able to change its sinking velocity quite significantly. And what I also want to talk about today a lot more is what are the potential mechanisms that various phytoplankton species might have to change this gravitational sinking. Now, our experimental setup is very straightforward. We culture isolates of a given species. We grow them in high nutrient conditions, which would be typically L1 media, or we grow them in low nutrient conditions, which would be L1 media where nutrient levels are made 100-fold lower, or where the L1 media is lacking specifically nitrogen or phosphorus or silicate in some cases. And then we just give the cells 5 days of growth. At the end of it, we have proliferating cells under one condition, we have these starved cells in another condition. We can do our mass and volume measurements, determine the average population densities and volumes, and put those into Stokes law together at the sinking velocity. And if we do this for this is like 9 species that we've done this for now, what we'll see on x-axis here is the relative sinking velocity change between low and high nutrient conditions. And what we'll see is that most species actually increase their sinking velocity when they are being starved. Now, there is one exception. There is a diatom that we found that decreases its sinking velocity quite a bit when it's being starved. So I want to talk a little bit about the mechanisms of decreased sinking velocity in that species. And then I'll talk a little bit more, especially with the green algae, how are cells able to increase their sinking velocity. And I want to note that we did not observe any meaningful shape changes. So while there are volume changes, the actual shape of the cells isn't really changing between these conditions. So the first thing we can do to break down what is actually happening in the cells and how is their sinking velocity changing when they're being starved is that we can just look at the cell volume and cell density measurements that we've carried out and separate them. So in this graph on the left-hand side of the screen, you'll see as a background coloring is the sinking velocity, and it's plotted as a function of cell density and volume. And the white dots that you see are the high nutrient conditions for any given species. Like if you see my mouse, there is the Chlamydomonas species, high nutrient condition shown as a white dot, and then the arrows are pointing to where the cells end up in if we starve them for given nutrient. So yellow arrow would show the nitrogen starvation, for example. And what you'll see is that, first of all, a lot of the arrows are pointing upwards, meaning that a lot of the cells are increasing their density when they're being starved. There's really one exception, that is E. Huxley. E. Huxley only increases its volume when it's being starved, and the increased sinking velocity in E. Huxley is purely driven by volume changes. Second thing we'll see is that especially if we look at the green algae, Chlamydomonas, Tetraselmis, and Dunaliella here, these arrows are pointing very much upwards. So these species seem to have a sinking mechanism that regulation of sinking that seems to rely more on density changes. Now we can quantify the influence that these volume and density changes have on the sinking velocity changes that we observe. And especially here shown in green box is the green algae species. So they are where we see most density regulation happening, and that influences the sinking. Now, it's worth noting that if you work with other model systems, you work with bacteria, yeast, you can starve them and they often get denser. There's some kind of quiescent state or spore-like state that the cells end up in, that's denser. But they don't get larger. So this is where phytoplankton seem to be kind of an outlier as model system to study, that most cells are getting bigger when they are being starved. So what's driving this? We see volume changes, we see density changes. How can we explain this in a slightly more mechanistic manner? Now, instead of trying to understand this on a level of a certain signaling pathway driving these changes, what we've done is we've tried to break apart the molecular composition of a cell. So we know that cells are very predominantly made of water, and then there's a lot of protein, and then depending on species, there's also lipids and carbohydrates. And these 4 components in most cases make like 95% of what the cell is made of. In species like Chlamydomonas, we actually have some estimates based from a literature on what is the relative composition of a cell. And if you know the relative composition, and then you know what is the dry density of a typical carbohydrate or what is the typical density of water, you can simulate how would a change in a given component change the sinking velocity of a cell. So what we have here is y-axis is the sinking velocity change that has been simulated, and x-axis is a fold change in that specific component. And for example, this specific simulation was done with starting values for species that doesn't have much lipids to start with. So even losing all the lipids in the cell barely influences sinking velocity because there isn't much lipids to start with. And under these conditions, if you really want to change the sinking velocity of a cell, what you want to do is you want to increase all dry content, because that would be mostly carbohydrate and protein, and both of them are very dense, so accumulating them would not only build volume, but it would really increase cell density. So this gives us some semi-quantitative framework of how to really think about changes in cell composition. And then we can go and measure these changes. And we'll start with the species of diatom that decreases its sinking velocity when it's being starved. And what we observe in this species is that this species, it contains quite a bit of lipids, and it's known to be a very high lipid accumulating species. So in our starvation conditions, in the low nutrient conditions, we actually see that in the electron microscopy pictures on the right-hand side, the lipid droplets inside the cell that are marked with the letter L, they can start taking up like nearly half the volume of a cell. It's like huge lipid droplets, and that's also very obvious if we label the cells for neutral lipids and do some fluorescence imaging as shown on the left-hand side. Now, the nice thing is that a lot of the lipid synthesis pathways are conserved, and we can use established inhibitors of lipid synthesis to partly prevent that lipid accumulation upon starvation. Now, it's only a partial inhibition, but it gives us some kind of toggle to study how much this lipid accumulation really matters for cell sinking. And what we see is that while in high nutrient conditions there's relatively little lipids, the lipid accumulation is more than tenfold when the cells are being starved, and we can remove about half of that with these lipid inhibitors. And this will cause a change in the measured sinking velocity of a cell. And in fact, if we correlate these changes, we see a fairly good correlation, which is really implying that at least part of the sinking decrease upon starvation in this species can be explained by the fact that it just builds up so large lipid reservoirs that the cell becomes lower density and starts floating or sinking more slowly. It doesn't actually float upwards. Now, it is worth pointing out that if we look at lipid accumulation in other species, the species that decreases its sinking velocity is the one highlighted here with the red square, excuse me. And it accumulates huge amounts of lipids, so of course that's going to influence its sinking. But we do have other species that also accumulate lipids when being starved. For example, Chlamydomonas species are well known to do this, and yet they sink faster when they are being starved. So it essentially highlights that there has to be other mechanisms at play that enable the cells to sink faster and counteract that lipid accumulation we see in species like Chlamydomonas. So what are they? Well, the obvious content would be water. And quite a nice tool that we've been developing in the lab is a kind of expanded version of the suspended microchannel resonator, where by tweaking the environmental media that you measure the cells in, you can measure different physical properties of a cell. So one thing we can do is that we can weigh our cells in normal media, that would just be essentially L1 media based on artificial seawater, but we can make that into heavy water, so D2O. And heavy water exchanges readily with the normal water inside the cell, so we'll get two buoyant mass measurements where one of them, the cell is surrounded by heavy water, so it's dense media, and the water inside the cell has changed too. And the other one is just normal water. And this allows us to really separate what the buoyant mass measurements are telling us. And the neat thing is that if you break down your buoyant mass equations so that you have this first term on top describing how the dry components of a cell influence your buoyant mass, and then you add to that how the water inside the cell is influencing your buoyant mass, then the water inside the cell has nearly the same density as the fluid outside the cell. You know, it's water, and it's water with a little bit of salts. That's the difference. So the latter term here approaches zero, and our buoyant mass measurements are very much reflective of the dry contents of a cell. And with this heavy water measurement, when we have two measurements, we can then solve for what is the actual dry volume of a cell. And if you know the dry volume, and then you just do total volume measurements, you can actually measure the actual water content of a cell, both absolute and fractional. So this is what we've done for not all the species, but a handful of them. And we see that the fractional water content can actually change quite a bit. We have examples like some green algae where the water content can drop more than 20% when the cells are being starved. And these are very exciting to me because it might reflect differences in the cell's cytoplasm. It might get very crowded in the cytoplasm. But I have to highlight that our measurements are agnostic to where the water content change happens. So you could also decrease your fractional water content by just building a really thick cell wall, and that cell wall has very little water, so the cytoplasm doesn't have to necessarily change. And quite surprisingly, if we take these water content changes and we then try to estimate how much they really matter for the sinking velocity, we find that they really don't. So in all the algae that we've looked at, what happens is that the cells are always accumulating a lot of dry contents. And it's those changes in dry content that drive the sinking velocity changes that we've measured. So whatever is happening with water is just not significant enough to change the sinking of a cell. And one way to think about this is that you have a small cell and it grows. When it wants to sink more, it's starved. Maybe it doesn't want to, but it's sinking more under starvation because it's grown larger. It has simply built a lot more dry mass than it has added water, and therefore the fractional water content might be lower, but the sinking change is really driven by the accumulation of dry mass. So water regulation is not the meaningful or not the key mechanism for the sinking regulation, the buoyancy regulation that we see. So what is it? We've looked into protein content regulation, but that doesn't seem to be the case. We do see changes in protein content in a lot of the species, obviously, especially when they are being starved for nitrogen. But those changes are never ones that can explain why sinking increases that much. And they are usually relatively small, like cells never lose 90% of their protein content. They probably couldn't survive at that point. Instead, if we focus on green algae, what we see is that there's a lot of carbohydrate accumulation. More specifically, these are the green algae species that we've been studying, and on the top layer here you're going to see high nutrient conditions, and then the bottom images are the starved low nutrient conditions. And these white bleps or granules that are appearing, these are starch granules, and they are considered to be very dense. So not only are they taking up a lot of cytoplasmic space and accumulating vast amounts, but because they are really dense, they are driving up the density of a cell. And if we just take our estimates of how much is starch accumulating and we put it into our simulations of how much would that actually matter for the sinking of a cell, we can largely explain the sinking changes in green algae, at least some of the species. So starch accumulation is really a key mechanism driving this increased sinking in green algae. We've also looked in some of the green algae, the compositional changes a bit more broadly, and really the cells become like 50% carbohydrates when they are being starved under low nutrient condition. And something I should have mentioned earlier, but I didn't, and it's going to be obvious now, we grow our cells in autotrophic conditions, so there's no carbon sources in the media. All the carbon comes from the light and photosynthesis. And so if you take away the light, there's no sinking velocity change, even when you try to starve these cells, because the cells just can't make these carbohydrates anymore. So I'm almost out of my time, but I think I can run through some conclusions and key questions. I have this kind of mental model of what's happening, and I apologize that this schematic here is going to be a little bit misleading. What we're really trying to illustrate is that phytoplankton might have a large set of mechanisms available for them to regulate their buoyancy and their sinking velocity, and this can influence where they position themselves in the ocean. Now, the cartoon is a little bit misleading in the sense that it shows this kind of idea that cell might sink from surface ocean to this border of the photic zone, and that would be like 100 meters. And I don't think that happens with any of the species we study, because the sinking velocities for the small cells are simply too slow. What is more realistic is that the cells might be moving up and down in the water column for, I don't know, maybe they move 5 meters or 10 meters. And that's enough to provide those cells with some degree of competitive advantage against their competitors, so that when they are low on nutrients, they try to sink a little bit deeper where they might have a little bit more nutrients available. And of course, motility might play a role in here, and we are only now starting to study motility. So we see that most phytoplankton increase their gravitational sinking when they are being starved, and there's multiple mechanisms that can influence gravitational sinking. A curious hypothesis that we can formulate based on our data is that this accumulation of carbohydrates may also not only make the cells sink more, but it might also function as a useful energy storage if the cells sink out of the photic zone, where they may not have as much carbon available because they don't get light anymore. And part of the reason I wanted to talk about this research at this audience is that I think we're seeing a lot of very extreme cell size and density phenotypes. Sizes and densities are changing way more than what you can easily find in a lot of other model systems. And this could be quite useful for our field of size and density regulation more broadly. And I also want to highlight one important, I don't want to call it shortcoming, but a key question that is still open here. Because we don't know if evolution has actually selected cells to regulate their sinking. So it could be that the cells are not aiming to sink more when they are being starved, but this could just be an accidental byproduct of other starvation responses. Now, whether that's the case or not remains an open question, but I think there's a lot of meaningful implications that come from this, and we're trying to study them. Something we're working on right now is how is motility coupled with the gravitational sinking velocity changes. Kind of an obvious question. I'll be happy to talk about that to you in maybe a year from now. And what I'm very interested in is that can we model the vertical movement and positioning of phytoplankton in situ based on the size and density measurements? Because if we can, we could actually start to address a little bit better whether the regulation of cell size and density is relevant to these ocean carpet and nutrient fluxes. And part of the reason I'm interested in this is that as a field of cell size researchers, we are very interested in our own specific topic, but this might help us plug into some, if I may say it, more important consequences of why cell size regulation matters. And I want to highlight that the size and density changes that we see doesn't have to act through the sinking of single cells, because those size and density properties would also be translated to any kind of cell aggregates, such as marine snow, that might be forming from this phytoplankton. So with that, I should highlight that there's been a lot of people involved in this work, especially Yanxi Wu, who's the first author of our paper. Everything I've shown you is out by now in PLOS Biology. And we've also had some great students, V and Thomas especially, working on this. So with that, phytoplankton are really pretty. If nothing else, that's why you should study them. And I welcome if you have any questions.
Amazing. Thank you very much, Teemu. And again, as before, just please post any questions that you have in the chat, and we'll go around one by one asking people to to read out their questions. So we can start with Kiki again. Would you like to read your question out?
Earlier, you had a plot comparing different species with each other, where I think one axis was density and the other was volume.
It was sinking and volume, yes.
Yes. Why does your plot not include any negative that is upward sinking velocity?
Yeah. We have never in our lab conditions found upwards floating cells. We've tried. So this is a very, very fine question. First of all, my speculation is that one would have to. My speculation is that it's not something that most cells are capable of. If you think about what a cell is made of and what would have to happen to cell composition for the cell to become buoyant and start floating upwards, it's really radical changes. Like you can swell up the cell 2 or 3-fold with water, and for most cells that won't do it. It just doesn't do it. And if you dilute everything 2 or 3-fold, like things don't work that well anymore. So you would really have to swell huge amounts, like Manu Prakash's work has shown in some species, or you would have to accumulate huge amounts of lipids that would be really low density and maybe get rid of some of like your carbohydrates and have minimal amounts of protein in cell and so on. And okay, go ahead.
But does this mean that these cells simply never travel upward?
Yeah, no, this is a really good question, and I've talked to ecologists about this. And honestly, the short answer is that we're kind of not sure. So obviously, a lot of the cells have motility. And as far as we understand motility regulation, a lot of it works based on phototaxis that makes the cell swim upwards. But a lot of cells, there's also plenty of species that are not motile. So are they purely reliant on upwelling of ocean currents? Or what's really happening there? We don't know. And like I've tried to talk to like ecologists modeling how particles are sinking in the ocean, and like the most honest discussions end up in kind of question marks.
Presumably, whatever the mechanism is, it would have to happen once a day.
Well, not necessarily. Like you're thinking of the DL vertical migration
Yes.
where cells go up and down. Yeah. So DL vertical migration certainly happens in some phytoplankton, but it's mostly a feature of zooplankton. I don't think it needs to happen once a day for phytoplankton. And we can again take the paper from Prakash lab with this, whatever the species was called, Noctiluca, that can swell huge amounts. And there it's about once a week type cycle. Yeah, so I think the once a day cycle makes sense, but there's just so much diversity in the species in the ocean that I wouldn't put in that assumption for any given species without knowing more.
Understood. Two of three, as you're going to see. Could measurements of sinking velocity be dependent on the time of day at which the measurement is made?
They are, yes, excellent question. So not all, but many of the species, their cell division is coupled to their circadian cycle. So basically, like you study something like Chlamydomonas, cells divide typically at nighttime, and they grow during the day when light is available. And then, because sinking scales with radius squared, if you measure the cells in the morning versus evening, the cells will be different size, and therefore the sinking velocity is different.
Did you control for that?
Yes, but not like perfectly. Like we try to do all our measurements between like 10 and 2, roughly speaking, and never do measurements in the evening. But like we didn't do every measurement within a 1 hour time window because just practicalities. The good thing, if I may, the good thing is that we've in multiple species we've looked at the magnitude of like that time-dependent change, and the starvation changes are so big in most species, especially the density change, that like you don't see that with the circadian cycle. Yeah.
I had a related question, which is, in these stress conditions, do you see any changes in cell division rates? I know you explained that they're typically coupled to circadian clocks, but when you remove nutrients in lots of other species, there are quite kind of defined programs that change the cell cycle to arrest them, right?
Yeah, so I should have been more clear about that. The starvation conditions are essentially no proliferation conditions. They are not like slow growth conditions. We really starve the cells. Okay. And that's almost like a control experiment we do, that we see minimal proliferation. Sometimes there's a little bit, but like we are comparing proliferating and non-proliferating.
So presumably that's how they're able to accumulate so much carbohydrate, because they don't divide, but they can still do photosynthesis.
Exactly.
Right?
Exactly.
Okay, yeah, that makes sense. Sorry, I might have cut off Kiki. Did you finish asking your questions, Kiki? You had a good list of them. I didn't want to.
My third one, I thought that there was a person between my first two and my third one.
It's okay. You go for it. I can read out Ben's question after, because I think he's left the call.
Yeah.
Understood. 3 of 3. You talk about the accumulation of carbohydrates, which are essentially energy storage substances, to promote sinking in response to starvation. However, this seems to mean that cells have the greatest need to accumulate carbohydrates when in a state in which they can least afford to divert energy for such purposes. Could cells get stuck in a state where they need to sink but don't have enough energy left to make the carbohydrates required to sink.
Right. Great. So let me clarify. The starvation is for nitrogen or phosphorus. So the cells will always, because they always have light, they always have carbohydrate production available, and essentially they have energy. So the starvation I'm talking about is starvation from nitrogen, phosphorus, silicon, depending on species. Or the low nutrient condition that we have is just all of those have been reduced at the same time. Now, so there's really never a situation where the cells don't have energy in our experiments because there's always light available. But there's maybe a related interesting question that why are the cells making carbohydrates and not lipids? Those two storage molecules for essentially carbon storage would have completely opposite effect on cell sinking. And why choose one over the other? And I don't know. I want to speculate that it could be about regulation of cell sinking, but that's purely speculation. I can't prove that at this point.
Thank you for your patience and knowledge.
Thank you.
I think Mimi wants to jump in.
You are muted.
Mimi.
Yes.
Sorry. Before Matthew/Ben asked the question, basically I was kind of jumping on, thinking along very similar lines to Kiki. Basically, like as these cells sink, right? Like they are also if you were in the wild in the ocean, you're going to also lose more light.
Correct.
So there's basically some sort of Pareto front like trade-off between the nutrient. Yeah. So I guess.
So in that sense, it could make sense to have that carbon storage as you're sinking in the low light environment.
Yeah. So then do you think that basically as you sink, you get slightly more, is the idea that you get slightly more nutrients, but then you lose light, so then you float back up? Like, is that how you think this
that's how I think it. Yeah, I can't prove that. Like, mimicking that is really surprisingly tricky in the lab.
I see.
But what I can tell you is that if you take, for example, the starved cells and put them back to higher nutrient conditions, they do revert back. These are alive cells, and when they start proliferating, they'll lose those excessive carbohydrate storages.
Yeah.
And they again decrease their density. They don't start floating upwards. We never see that, but at least with the green algae, they are all motile. So maybe they just have a upwards motility driven by actual swimming, and whether that upward motility by swimming is coupled to the other sinking behavior, we're looking into it right now.
Yeah. Lots of mammalian cells also store, glycogen in this exact way. You have these not starch but glycogen granules that people don't know. Yeah, it completely changes their density. It's interesting.
Yeah.
Yeah.
I guess connected to this, I can ask Ben's question. Ben apologizes because he had to join another seminar immediately after this one, but he asked how constant and uniform is cell density for a given environmental condition? Can any of the cells actively modulate on behavioral timescales? Which I guess, is of course connected to the same.
Yeah, it's a fine question. First of all, we have in few species looked into single cell densities, and they vary much more in phytoplankton than they do in mammalian model systems. But this is kind of hand wavy, what is a lot of variability, what is little. I can just say that it's more than what we see in whatever budding yeast or mammalian cell culture cells. There's more variability in density. Now, how much can that be modulated on behavioral timescales? In the couple species that we have data, and they are green algae species mostly, during the day density doesn't change, except we see it change in Chlamydomonas. So, I mean, maybe I just have studied too few species, but it looks like the kind of workhorse of phytoplankton is the outlier. Here, let's see, and there it changes in kind of counterintuitive way. In a sense, the cells seem to be getting denser through the day. So not only are they getting bigger, but they're getting a little bit denser during the day, and therefore the sinking difference, depending on time of the day, is quite big. But that's all very preliminary. We don't know too much.
So Jan has a question.
Yeah, Teemu, I was wondering if you grew them in media of different density, does it somehow respond to, I don't know if there's a way of
Yeah.
like changing the density of the media, like does it in
Yeah.
some way know that it's sinking?
We haven't done that, to be clear. My guess would be that I don't know how cell would know that, except
Yeah.
through cues in. So if I think about how can a cell know its vertical position in the ocean, it might react to nutrients, it might react to light, but both of those are location dependent. Like those
No,
are
but there
things
may
that
be a velocity sensor. If it has some appendage and it's flowing, it's gonna produce some
that would be fun. Yeah,
force.
yeah. Yeah, you're right. That could work. We haven't done that. My favorite hypothesis is that if a cell wants to know where it is depth-wise, it doesn't know it wouldn't be a velocity sensor, but it would be a depth sensor. The only universal factor that cell would sense is pressure. Like pressure is the one thing, and like you gotta remember that like it's not clear to me how phytoplankton couple all of these things together because like you think about phytoplankton, there's tons of diatoms in like Arctic oceans, for example, and they don't get light for like a few months of the year. So I don't think they set their sinking velocity or sinking depth purely based on light. But pressure would be like the one universal factor that if cells could sense, that would be cool. But so far I haven't found any MIT undergraduate who would build me like a pressure cooker that we could actually have in the lab according to health and safety rules. And yeah,
Yeah, those are dangerous. They can also be bombs.
and you would think that kind of gets some people excited, no?
I would think it would get them excited, but I could also see the
Yeah.
concern of the safety officer.
No, it's fine. We'll wear safety goggles.
Safety goggles, yeah.
Yeah, we'll put on gloves. It's safe. It's fine.
Well, on
Yeah.
that
No.
inspiring note, I think we've come to the end of the list of the questions. I'd like to thank Ben and Teemu again for two really fun and enjoyable talks, and we look forward to seeing everybody again in 2 weeks' time for another set of speakers. Thanks, everyone.
Thank you.
You.